This is Module 5, Session 2, where we're going to look at some molecular techniques used in diagnostic evaluation. This is synergistic with Chapter 17 in your textbook. In general, we'll discuss some molecular techniques. The basis of molecular biology utilizes the structure of DNA and the naturally occurring enzymes to allow for the generation of various in vitro systems. We're also going to look at the use of diagnostics. The utility of these techniques is several-fold. They can be used to determine genetic changes at the level of DNA, RNA, and protein. They can also be used to generate therapeutics, recombinant proteins, and other viable technologies. So first off, why use molecular biology techniques? Gene mutations can have serious consequences. As we've seen in several modules, a defect or a loss of an enzyme can cause serious symptoms and phenotype changes. We can use molecular biology techniques to track these gene mutations and to try and predict heritable changes. This helps us predict a risk of disease. Molecular biology techniques can differentiate between normal genes and mutations. It can also help identify what type of mutation is present. This may impact the therapy chosen. Likewise, these techniques can help us develop new recombinant therapies, such as gene replacement or protein replacement therapies. Let's start with plasmids and their use in gene cloning. In summary, plasmids are extrachromosomal DNA molecules. They're circular in nature. There are three basic requirements of plasmids. First off, they have to have the ability to self-replicate within a host. They require an ORI, or an origin of replication. They usually need to have a selectable marker, meaning you need to be able to identify whether the cells you're looking at have or do not have the plasmid of interest. Second, they must be capable of being digested, meaning they must have cloning sites that include restriction enzyme sites, which we'll discuss in the next slide. This is a schematic of a general plasmid containing an origin of replication, a cloning site where you can introduce your gene of interest, and a selectable marker, so you can identify cells that have the plasmid that you're working with. In the multiple cloning site region, there are restriction endonuclease sites. These are important tools for recombinant technology. In summary, these are enzymes that recognize short 4 to 6 base pairs sequences of DNA and cleave the phosphodiester backbone. They are named for the bacteria they are isolated from. This schematic shows DNA digestion by the enzyme wk05. The cut site or restriction site for wk05 is shown here. On the 5' to 3' strand, the restriction site reads GAATC. The enzyme recognizes and cleaves the phosphodiester backbone between the G and the A on one strand and the G and the A on the complementary strand. This leaves what is known as sticky ends, where there is an overhang on both cut fragments. Sometimes restriction enzymes leave blunt ends, where the cleavage, for example, is straight between two base pairs. In this case, there is no overhang, like this, left after digestion with that restriction enzyme. You can use both plasmids and restriction enzymes for cloning. Cloning combines the utility of both of these molecular tools. When the vector and DNA, the vector being the plasmid and DNA would be your gene of interest, are digested with the same enzymes, these can be joined together using a DNA ligase. This is similar to the DNA ligase used in DNA replication. You can create this environment for DNA ligase to be active in an in vitro setting. DNA ligase will then catalyze the formation of a phosphodiester bond between these two complementary fragments, resulting in recombinant DNA that can be introduced into cells. Let's look at that here, where you have two different DNA fragments digested with EcoR1. EcoR1 produces sticky ends, shown here and here, that have complementary overhangs. Mixing of the two DNA fragments with the addition of DNA ligase will allow for the fragments to be ligated together. Ligation of this fragment in vitro is considered recombinant DNA. You can use this technology for the production of DNA or protein in bacteria. If you start with your plasmid and DNA to be cloned in, or a gene of interest, you can digest them again with the same restriction enzyme. The fragments shown here can be ligated into the plasmid, which will now contain your gene of interest. This can be inserted or transformed into bacterial cells. If we were using eukaryotic cells, the term would be transfected. The bacteria can then be selected for with our marker on our plasmid, such that we can grow bacteria that have our plasmid of interest. When you grow large cultures, you can do one of two things. You can harvest the culture to obtain DNA by isolating the plasmid from the bacteria. Once the plasmid is isolated, you can digest again with your restriction enzyme of choice to remove the cloned DNA. In this manner, you can generate large amounts of cloned DNA. Alternatively, you could use these bacteria to express protein. The plasmid often has a gene promoter which can be used to promote transcription and translation of your gene of interest. In doing that, you can isolate large amounts of protein from these cultures. This is a manner in which insulin is expressed recombinantly. This is an artificial means for generating large amounts of insulin for therapeutics. It's a way of replicating a missing protein. If you know anybody who has type 1 diabetes, you know that they are lacking insulin production. Consequently, they must give themselves insulin exogenously. Recombinant insulin is made in this similar manner, where the insulin A chain and insulin B chain are in two separate plasmids. These plasmids are transformed into E. coli and grown in culture. The protein is then expressed and purified, and the two proteins are mixed and refolded to allow for cysteine bonding between the two polypeptide chains to generate an active insulin. Polymerase chain reaction is another molecular technique for generating large amounts of DNA. It amplifies DNA molecules exponentially using DNA polymerase. You only need to know the sequence of the flanking bits of DNA to design primers to initiate the replication. Primers are short oligonucleotides representing forward and reverse sequences. These are similar to the RNA primers required for normal DNA replication. In either case, each primer anneals to the 3' portion of the DNA template. Let's look at this here. In this example, you have a DNA double helix shown here with a region of interest to be amplified, highlighted in yellow. In the first step, you can heat the DNA, which allows for denaturing of the DNA into single strands. Once the DNA is denatured, you cool it slightly, and the primers, shown here in blue, will anneal to the single-stranded DNA. Once they're annealed, DNA polymerase will synthesize a new strand of DNA. This reaction can then be repeated, where there's heating and cooling, shown here, where the single-stranded DNA can then bind again with a set of primers and be amplified again. The cycling of these reactions of heating, denaturing of DNA, binding or annealing with primers, and replication with DNA polymerase allows for rapid amplification of DNA. All in all, 4 to 20 cycles will represent 10 to the 6 copies. Once you've replicated your DNA or isolated your plasmid from bacteria, it's always good to look at your DNA sample on gel. This gel for DNA consists of an agarose matrix. If we remember, DNA is negatively charged and will run to a positive pole, shown here. Small molecules will migrate further in the gel than larger molecules. DNA is separated by both charge and molecular weight. If we stain the gel after the addition of our sample, in this case, you can see multiple bands that are separated based on molecular weight. In addition to cloning and PCR, there are several procedures referred to as blotting procedures that can help identify changes at the level of DNA, RNA, and protein. At the level of DNA, large expansions or deletions can be visualized by a southern blot. Large deletions or expansions are common in triplicate repeat disorders such as Fragile X or Huntington's disease. In the case of Fragile X, a southern blot is used to identify the presence of triplicate repeat expansions. A southern blot looks at changes in genomic DNA probed with a DNA fragment. At the level of RNA, you can examine changes in the amount or the size of a messenger RNA through a northern blot. A northern blot consists of looking at total RNA and probing with an RNA or a DNA fragment. Finally, protein and changes in the amount or the size of protein can be determined through a western blot. A western blot looks at total protein probed with an antibody specific for your protein of interest. Let's look at each of these now. Before we do that, to clarify, a probe, in all cases, is a labeled fragment that anneals with high level of specificity. In this case, we're looking at a probe that hybridizes with single-stranded DNA. This allows us to visualize only the strand of DNA that the probe is annealed to. This is true for all of these blotting procedures. The probe, in the case of a southern blot, is DNA that will hybridize to genomic DNA. In the case of a northern blot, the probe may be DNA or RNA that hybridizes to an RNA strand. In the case of westerns, the probe is an antibody that is specific to your protein of interest. In all cases, when you visualize these experiments, the only thing you will see are fragments in which the probe has identified. Let's look at a southern blot. This compares southern, northern, and western blotting. In the southern blot, we're looking at total genomic DNA. The DNA is run on an agarose gel and transferred. At this point, the bands are not visible. Before probing, you can visualize the bands by the addition of a dye that will bind DNA. Once the probe is added, which is usually fluorescently labeled or radioactively labeled, this will identify our bands of interest, shown here. Similarly, in the case of a northern blot, total RNA is run on a gel. It's transferred and is not visualized until you've stained to identify all of your total RNA. Once the probe is added, in this case a DNA probe, we can then identify only our bands of interest. Finally, a Western blot, where you are looking at a protein of interest, you'll load a total protein lysate that can be visualized with a general stain, then probed with an antibody specific to your protein of interest. Let's summarize the previous techniques by looking at this question. Which of the following statements is true? Restriction endonucleases are not required for insertion of your gene of interest into a plasmid. Choice B. PCR is only an available technique if you know the full sequence of the region of interest on a stretch of DNA. C. Southern blotting evaluates changes in the size of mRNA. Or finally, bacteria can be used as viable hosts for both protein and DNA expression. Let's start with A. This is a false statement. Restriction endonucleases are required for insertion of a gene of interest into a plasmid. The presence of multiple cloning sites is one of the primary resources required by a plasmid. The other two, the plasmid must have an origin of replication and a selectable marker. An example of a selectable marker may be a gene encoding for antibiotic resistance. B. This is also a false statement. PCR can be used, and you only require knowledge of short, flanking regions of DNA, short stretches, 15 to 20 nucleotides, so that you can generate homologous primers that can bind the complementary stretch of DNA. It is not necessary to know the sequence of the full region of interest that you're looking at. Choice C. Again, this is a false statement. Southern blotting, remember, looks at changes, large changes, insertion or deletions in DNA. It does not evaluate the size of messenger RNA. That leaves choice D, which is true. Bacteria, when transformed with the appropriate plasmid, can be used for expression of both a protein of interest and DNA.